In this lecture, we're going to focus on the transmission of information along the neurons. So our objectives will be to describe the membrane potential of a resting neuron, explain how the sodium-potassium pump helps to set up the resting membrane potential, then diagram and explain the initiation and propagation of an action potential, explain how myelin sheaths increase the speed of the action potential, and describe a synapse and the movement of a neurotransmitter across the synapse. We're going to start with a big picture view. Along a neuron, the signal is electrical, movement of ions, and we call it an action potential. Between the neurons, the signal is chemical with neurotransmitters, and it's happening across this gap in our synapse. As we start talking about an action potential, we're going to look at diagrams, we're going to draw sketches, and I want you to pay particular attention to the creation of a charge differential across the cell membrane of a resting neuron, the function of the sodium-potassium pump in creating that charge differential, how depolarization initiates a nervous impulse, and how that helps us to understand why the signal goes in one direction, in dendrite out axon, and why the signal is all or nothing. Once the signal starts, it goes. This is organized in questions so that we're really focusing on the big picture. Your book has a lot of detail, but what I really want to do here is give you that big picture, and then you go back to your book and you add in some of those other details. So in our resting neuron, our resting membrane potential, the cell is relatively more negative where? And the cell is relatively more positive where? And what membrane pump is helping to set up this gradient? So it's an electrochemical gradient. And I see that it's relatively more negative inside, so more negative charges inside, and more positive outside. So more positive charge on the outside of the membrane. And the membrane pump that's helping to set up this gradient is our sodium-potassium pump. For every 2 potassium, we're going to pump out 3 sodium. And that's going to help us to set up that gradient. More negative on the inside, more positive on the outside. And you might be thinking at first, wait a minute. They're both positive ions. How are we getting positive on the outside, negative on the inside? Remember, I keep saying more. It's a relative charge. So if for every time those pumps are going, you're pumping 3 positive charges out for every 2 positive charges going in, then you are getting more positive charges building up on the outside. It gives us that relative difference. It's not even a very big difference. It doesn't have to be. So what starts an action potential? We just saw what a resting neuron looked like. Resting neuron, we have relatively negative charges inside, relatively positive charges outside. What starts that action potential? And what we need is a strong enough stimulus coming in the dendrites and the cell body to open up another protein in the membrane. And this protein is our sodium gate. So what starts an action potential? Our sodium gate opens and sodium diffuses into the cell. So now I've got sodium diffusing into the cell. So that means a lot of positive charges are coming in. And what happens is that builds up relatively more positive charges inside, and now the outside is relatively more negative. That's what we call depolarization. When we switch from being more positive, more positive on the outside and more negative on the inside, and we switch to more positive on the inside and more negative on the outside. And what happens there is this will cause another sodium gate to open, and in comes the potassium or sodium, and that causes our depolarization again, which is going to open up another sodium gate. And in comes the sodium, which is going to cause depolarization. And then as soon as the message passes, we have yet another channel opening. This is our potassium channel. And potassium will diffuse out. So potassium diffuses out. And that's going to give us a lot of potassium now on the outside of the cell. There are some other parts of this process, but I'm keeping big picture here. So then we get a buildup of potassium out here, sodium in here. Our sodium-potassium pump goes into action, pumping the potassium back in, the sodium back out, and that's all part of repolarization. So the opening of the potassium gate, and then the action of the sodium-potassium pump helps to repolarize or put that neuron back into the resting state. Okay, so that was big picture. Again, get that down first. Then go back to your book and look at some of the other details. And hopefully that will help those details make more sense. For our action potential, once they're started, they go. However, myelin sheaths can help the axon or the action potential move faster. So myelin sheaths, remember, that's where you had your Schwann cells wrapped around the axon, making like a fatty insulating covering, or the oligodendrocytes in the central nervous system. One thing they do is help the cell to conserve energy because it helps to kind of cut down on leakage of sodium and potassium across the membrane. Even though they're polar, they do leak a little bit. So helps to conserve energy of the sodium-potassium pump. But also very important is it speeds information transmission. So saltatory means jumping. Saltatory transmission is where the signal jumps from node of Ranvier, to node. So you can see in this diagram here, we have our sodium gates opening, sodium pouring in. That's going to cause depolarization. And then that signal jumps to the next node, opens up those sodium gates. That's going to cause depolarization. Then that signal jumps to the next node. It's the difference between something along the order of 250 miles per hour rather than 5 miles per hour traveling. So a big difference in speed. And myelin sheaths, particularly the Schwann cells of the peripheral nervous system, can help regenerate damaged PNS neurons, not CNS. Oligodendrocytes don't work this way, but they create a tube so that if something happens to the axon, a new axon can grow out and follow this tube. So our speed of signal is dependent on myelin sheaths, the presence of myelin sheaths, faster, absent, slower, and also on the thickness of the sheath. The more wrapping, the faster the signal. The less wrapping, the slower. Also depends on the diameter of the axon. A wider axon transmits that action potential more rapidly. With synaptic transmission, pay particular attention to the synapse, the release of the neurotransmitter, and the binding. So the information is coming down the axon here of our presynaptic cell. And when that action potential reaches the presynaptic axon terminal, it's going to cause the opening of these gates and also the release of neurotransmitters. So here's our neurotransmitters. So neurotransmitters. going to release neurotransmitters in this cleft. So there's a space between the presynaptic neuron and whatever it's talking to, whether that's the dendrites of another neuron or a smooth muscle cell or a skeletal muscle cell or a gland cell. There is a physical gap. They are not touching each other. The neurotransmitters will diffuse across that gap, bind to the receptors here, and start initiating a response in our postsynaptic cell. And this is usually a graded response, meaning you have to get enough bindings of neurotransmitter to get enough of our signal so that we can actually get a response in this particular cell. So, structure of synapse, physical gap, release of neurotransmitter from presynaptic, binds to the postsynaptic, and then after that binding, we have enzymes that break down the neurotransmitter, and then the pieces are brought back up into this presynaptic cell and used to make more neurotransmitter. Surviving the nervous system, it tends to be one of the harder chapters. So the nervous systems can be a very complicated subject, and here are some survival tips. Don't be afraid to diagram in color. Notice how I left spaces so that I could diagram as I was going. Get active. Don't just look at the picture. Don't just read the information. Draw it. And make sure you have the big picture relationships first, then add detail. This lecture was focused on what's a resting neuron and what's the resting neuron membrane potential. What are the main steps in an action potential? What is a synapse? How do neurotransmitters move from one neuron to another? You do that first, then details. And get on the internet. Use animations. They are so valuable. I would have loved animations when I was in college. I would have done things that probably aren't even legal for them. Okay, so good luck, and if you've got questions, make sure to contact myself or Dr. Fisher, and I will see you in the next presentation.